Yes, I'm Aleksandra Nijak, and I'm happy today to present to you the results of my PhD thesis in which I used iPSC-CMs to model Brugada syndrome. I will start my presentation with a short introduction of the topic and stating the aims of the thesis, and I will move forward to discuss the results from three chapters of the thesis. And I will finish the presentation with a short discussion on future perspectives for Brugada syndrome research. Every cell in the human body contains DNA, genetic information, in a form of a string of nucleotides, which forms a DNA. And the DNA is then condensed into chromosomes, which fit into the small compartments of the cells. When the cell divides, DNA is transcribed into mRNA, messenger RNA, which is further read on ribosomes in trios, codons, which encode amino acids, which are further translated into a chain of amino acids that forms a protein. Although natural mechanisms exist to prevent happening of the errors in those processes, like, for example, existence of 2 copies of each gene in the DNA sequence, from time to time mutations can occur, which, when they change the amino acid sequence in the protein, can lead to translation of the mutant protein and contribute to the disease. So one type of the genetic disease is Brugada syndrome. It's an inherited cardiac arrhythmia which affects about 4 million people worldwide. The disease affects mostly young adults who present with variable phenotypes, starting from patients who don't experience any symptoms to those who experience syncopes and sudden cardiac death. The only existing diagnostic criteria are the presence of SCN5A mutation. However, this explains only about 25% of individuals, meaning that over 70% of the patients don't have genetic diagnosis. Another diagnostic criterion is the presence of the typical BRS type 1 ECG pattern, where we can observe a curved ST segment elevation. The disease was thought to be inherited in autosomal dominant pattern, meaning that a single mutation in SCN5A is sufficient to develop symptoms. However, because of the variable phenotypes and low penetrance of the variants, the disease now is thought to be inherited in a more complex way. SCN5A is the gene which encodes sodium channel proteins, which are expressed on top of the cell membrane of the cardiomyocytes. It would be then interesting to study the disease in biopsies from the hearts of the donors. However, this procedure is very rare and carries high risk of complications. Thus, in laboratory settings, we need to find another way to obtain those cells to study a cardiac arrhythmia like Brugada syndrome. For this purpose, we can obtain a simple skin biopsy or a blood sample from the donor, from which the cells are brought into culture and then further reprogrammed for using transcription factors into induced pluripotent stem cells, iPSCs. Those cells carry potential to differentiate into virtually any cell type of the human body. And in case of my PhD, I differentiated iPSCs into cardiomyocytes, iPSC-CMs. So to study the mutant sodium channel proteins, we can use a patch clamping technique, which is schematically represented in the figure. For this purpose, cardiomyocytes are placed in this bath solution in which also a ground electrode is placed. To close the circuit, an intracellular electrode is placed in a glass pipette which approaches the cell and seals on top of the cell membrane. And with this setup, an external amplifier is able to measure the flow of currents across the cell membrane. In this process, we obtain a graph of the sodium current in response to the applied voltage to the cell. Looking at this graph, we are able to study activation and inactivation of the channel, which means opening and closing of the channel, as well as the peak current density that flows across the cell membrane. In Brugada syndrome, the affected proteins are characterized by a loss of function observed as a reduction in the sodium current in comparison to the wild-type protein. Nevertheless, the cardiac cells express not only sodium currents and sodium channels. To study the function of all of the channels present in the cardiac cell, one can study action potentials. Those action potential waveforms were shown to be similar in iPSC cardiomyocytes to those measured from ventricular cardiomyocytes. In this waveform, one can look at the action potential amplitude, APA, or upstroke velocity, denoted as dV to dt max, which are corresponding directly to the function of the sodium channels. One can also measure action potential duration, as well as resting membrane potential. Another complementary technique to study electrical function of cardiomyocytes is measuring of the calcium transients using fluorescent dyes, where we measure the flow of ions visible as the fluorescence flow. The calcium transient waveforms are very similar to action potentials. However, this is the function of the calcium handling. And as this method is more high throughput and enables us measuring more cells than only single cell in action potentials, we can study the propagation of the electrical signal between the cells. This brings me to the aims of my thesis. First aim I wanted to answer was to unravel the genetic basics of Brugada syndrome. For this purpose, I studied 3 Brugada syndrome families in which the screening with the PET panel with known arrhythmia genes did not yield any genetic explanation. For this purpose, I wanted to see if mutations in novel genes or potentially copy number variation could explain the missing heritability of the disease, or if the answer lies maybe in the non-coding parts of the genome. Next, I aimed to model the disease in iPSC-derived cardiomyocytes, and here I looked if it's possible to model known Brugada syndrome SCN5A mutations in this cell type. Next, if it's possible to model Brugada syndrome in iPSC cardiomyocytes derived from patients who are genetically unresolved. And finally, if it's possible to use this cell model to study novel mechanisms of the disease. This brings me to the results section of my presentation, and I will start this section with discussing the results from the genetic screening of the 3 Brugada syndrome families, in which I used whole genome sequencing and linkage analysis. Linkage analysis is a technique in which one types several markers across the whole genome, and from those markers, one selects those that are inherited by all of the affected individuals within the family. In this example, one can notice the affected individuals presented in the full symbols are present in all of the generations. This suggests a genetic contribution to the disease inheritance. In the linkage analysis in this family, location on chromosome 3 with a specific genomic directives was identified, and it turned out it encodes, there is located a DNA sequence for SCN5A gene. In this presentation, I will discuss results only from one family that I investigated, for which I obtained the most promising results from the combined linkage and whole genome sequencing analysis. In this family, 13 individuals were used for the linkage analysis. They are indicated with green box. And 3 peaks were obtained on chromosome 2, 10, and 19, Those peaks were studied further in the whole genome sequencing data, which I obtained from the individuals indicated with the red star in the pedigree. I performed the step-by-step analysis of the whole genome sequencing data, first looking at the shared rare variants in the coding regions of the genome. I identified a variant in exon 2 of SNAP29 gene. This variant was predicted to be damaging to the encoded protein. However, due to the ubiquitous expression of the protein and involvement of the protein in the autophagy in all of the tissues in the human body, the variant was excluded from the candidate list. Next, I looked into the copy number variation, and here I identified 100 kilobase per deletion in intron 5 of a known arrhythmia gene, DPP6. However, the variant was excluded from the candidate list due to the fact that it didn't affect the splicing of the protein, nor was affecting a known enhancer or regulatory site of another genes expressed in the human heart. Finally, I looked into detail into the non-coding regions located under the linked peaks, and here I identified 2 intronic variants in 2 genes, one candidate, arrhythmia candidate gene, CTNNA3, and one in GLI2 gene, which is a known regulator of myocyte enhancer gene, MEFC2, and this gene is involved in embryonic development of the heart. Both of those variants had prediction to affect splicing of the affected proteins. Thus, I decided to perform segregation analysis in the family and noticed that both of the variants segregate with the disease phenotype. And because of this fact, I decided to perform splicing analysis of the variant in iPSC cardiomyocytes from the 3 individuals from the family indicated with yellow stars in the pedigree. As both of the variants were affecting intronic parts of the genes, I designed the primers for PCR analysis in a way so they span neighboring exons, and in the occurrence of an alternative splicing, in case a piece of intron is retained or an exon is spliced out, I could see differences in the sizes of the products on the gel electrophoresis. So to study the splicing of those variants, I obtained RNA from iPSC cardiomyocytes, which were cultured in a normal differentiation protocol and with incubation with an antibiotic, puromycin, which is a known blocker of a nonsense-mediated decay, a natural process in which wrongly transcribed mRNAs are degraded to not produce mutant proteins. However, after the analysis of my bands, I did not see in the patient samples any difference in comparison to the controls, meaning that most likely the splicing is, there is no effect on splicing based on the obtained results. During the course of the experiments, new information became available about one of the patients who was initially diagnosed as unaffected. This patient was shown to carry a variant of unknown significance in ankyrin2 gene, which is a known arrhythmia candidate. And the variant was absent in all other affected individuals in the family. Thus, I decided to include the patient and her mother into extended linkage analysis. And here, the peak on chromosome 2 became more significant. However, the analysis of the data from whole genome sequencing did not any new candidate variants in this peak. So far I did not find any candidate in my family, I decided to go back to the analysis of the whole genome sequencing data with the idea that maybe with the updated bioinformatic pipelines I could yield some more information. And In this process, I identified variant in exon 8 of DCLRE1A gene, which was predicted to affect normal splicing of the protein as presented here in the table. The gene was previously reported to be involved in case of a Fanconi anemia, where some patients present with congenital heart defects. Thus, the variant became a potential candidate. I performed a segregation analysis and saw that in the available affected individuals, the variant co-segregated with the disease phenotype. And the further splicing analysis of the variant will be carried out in the follow-up studies. In conclusion, I was able to identify a linked region on chromosome 2 in the tested family. I did not see the missing heritability in copy number variation. And I was able to identify a potential candidate variant in DCLRE1A gene. I'm sorry, this is a very difficult name to spell. So how is it possible that with the significant peak on chromosome 2, I was not able to find a potential candidate? It's quite possible that in this region we are missing some information that are not available in the databases. So it would be interesting to repeat the analysis of the region in a few years and to see if we have known something more about that region. Or another explanation could be that the common variation, which was not studied in this case, could be explaining the missing heritability in this family. Now I would like to move forward to discussion of the results from iPSC cardiomyocyte models of the Belgian SCN5A founder mutation. A founder mutation is a genetic alteration which occurs with a high frequency in a geographically or culturally isolated group, and it originated from a single or multiple donors and is present multiple generations later in more or less not changed form. The Belgian SCN5A founder mutation is a duplication of 4 nucleotides, which are located in exon 27 of SCN5A gene, and it leads to activation of an alternative splice site, and the product of the splicing is a 32 amino acids shorter protein in comparison to wild type. The variant was identified in over 25 families in the Antwerp region, and all of the affected carriers present variable phenotypes, starting from those who don't experience any symptoms to those who have a sudden cardiac death event. The variant was tested in transient expression model and was shown to lead to a loss of function of the NAF1.5 protein in a form of the reduced sodium current density. To study the variant, I obtained 7 clones from 5 donors, including 3 clones from the control donors. 2 donors presented with ajmaline-induced BRS type 1 ECG, and 1 donor presented with syncopes and an SCD. The cells were generated with multiple differentiation protocols. However, in this presentation, I will present results from the protocol in which we addressed maturation of the cells, adding T3 hormone into the cell culture media. I noticed a sodium current reduction in 3 of the tested clones. Those were all clones from asymptomatic donors who presented with ajmaline-induced BRS type 1 ECG pattern, BRS1 indicated in gray. I'm sorry, I think the battery is getting low. BRS1 in gray, BRS8 clone 2 in red, and BRS8 clone 3 in dark red. Interestingly, BRS8 clone 3 showed reduction of the sodium current only in comparison to one of the tested controls. Next, I looked at the voltage dependence of inactivation of the sodium current and noticed that in the severely affected donor cells, indicated in green, I noticed a negative shift of inactivation in comparison to the controls, which explains a loss of function effect of the affected channel, meaning that less channels are available in this cell line in comparison to controls. Looking at the inactivation curve from BRS8 clone 2, I noticed a positive shift in the midpoint of inactivation, which means a gain of function of the protein. Finally, I looked at the action potential properties obtained from the affected cells, and here I noticed action potential duration reduction in all of the tested patient cells in comparison to only one control cell line. Interestingly, no changes in upstroke velocity or action potential amplitude were noticed for all of the tested patient lines. So as I obtained cells from different donors with the different severity of the phenotype, I tested if I am able to pick up the differences between the severity groups in my iPS cardiomyocyte model. And here, interestingly, I noticed that in the more severely affected cells from the more affected patient indicated in green, BRS3 clone 2, I noticed higher sodium current density and the faster upstroke velocity in comparison to asymptomatic group. This could mean that there is any compensatory effect on the channel function in those cells in comparison to the asymptomatic group. However, looking at the sodium current inactivation curve, in the cells from the more affected patient, I noticed a negative shift in the midpoint of inactivation, which could explain the more severe phenotype in this donor. In conclusion, I was able to see loss of function effect on the sodium channel protein in all of the generated iPS cardiomyocytes. However, those phenotypes were showing variability. In some patients, it was just a loss of function effect. In some, it was a mixed gain and loss of function effect. Looking at the different severity groups, I saw somewhat unexpected results for sodium current density and the upstroke velocity. However, the more negative midpoint of inactivation in the more severe patient cells could explain the severity of the phenotype. So the hypothesis for the observed differences in the phenotypes could be that another variants existing in the patients could explain could modulate the phenotype observed in my iPSC cardiomyocytes and potentially the phenotypes of the patients. Finally, I would like to discuss the results from functional phenotyping of iPSC cardiomyocytes obtained from the genetically unresolved donors. For this purpose, I obtained iPSC cardiomyocytes from family members from one of the families which I studied in the previously discussed chapter. For this purpose, I obtained cells from BRS1, affected female donor with normal baseline ECG, and exercise or Ashmalin-induced BRS type 1 ECG, BRS2, BRS2, the affected male who presented with history of syncopes and Ashmalin-induced BRS type 1 ECG, and one unaffected donor which was serving as a family control. For those experiments, I also used two other controls, one negative unrelated control and one positive control who was a carrier of BRS SCN5A founder mutation. Looking at the sodium current properties, I noticed, reduction in sodium current density in BRS1, which is presented in the red boxes in the figures. And I also noticed positive shifts in the voltage dependence of activation and inactivation. Nevertheless, looking at the very low current density that I measured in the cell line, The observed shifts in the voltage dependence of activation and inactivation are possibly not related to the phenotype and are because of the high possibility of errors in the analysis. Interestingly, looking at the BRS2 indicated in the red circles in the figure, I did not see any changes in voltage dependence or sodium current density. Next, I looked at the action potential properties in the obtained cells, and here I noticed again in BRS1 reduced action potential amplitude as well as upstroke velocity in comparison to all of the tested controls, which goes in line with the observed sodium current changes. Again, in BRS2, I did not see any changes in action potential properties. And looking at the action potential waveforms, in both BRS1 and BRS founder iPSC cardiomyocytes, I noticed a loss of characteristic action potential dome. As I was not able to pick up the Brugada syndrome phenotype in BRS2 iPS cardiomyocytes, I decided to perform Ashmelin challenge on the cells to see if I am able to induce the Brugada phenotype in the action potential properties. And here, looking at the action potential amplitude and upstroke velocity, unfortunately, I didn't see any changes in my control cells, thus making it impossible to study the effect of the sodium blocker on the patient cells. Finally, I looked at the calcium transient properties in the measured, in the obtained cells, and noticed in BRS2 prolonged calcium transient duration and rise time, which was similar to what I observed in BRS founder. Moreover, looking at both recordings from both BRS1 and BRS2, I noticed higher percentage of early after depolarization in my recordings, with much higher prevalence in BRS2, which reached over 30%. And taking into account all of the results from the sodium current characterization, action potential characterization, and calcium transients, I hypothesized that in BRS2, it is possible that the disease could be explained by affected calcium handling. In conclusion, I was, on the contrary to what was previously published for iPS cardiomyocyte models for the genetically unresolved cases, I was able to see Brugada syndrome phenotype in my cells. And interestingly, I saw that there might be a potentially different disease mechanism in the two family members. The negative Ashmelin challenge result is not an uncommon observation. Previous reports on the Ashmelin challenge performed in iPS cardiomyocytes from genetically unresolved cases is gained the same results as me. They were negative. And the explanation for this observation could be the immaturity of the cells, which is a known drawback of this model. However, as I was able to pick up the Brugada syndrome phenotype in my cells, it probably would be interesting to look more into detail into RNA sequencing to see if we can find in this way the genetic explanation for those patients. So what does the future hold for Brugada syndrome research? In my opinion, based on the collected evidence and based on the newest publications in Brugada syndrome research, I believe next to search for rare mutations and rare variants, maybe it's important to also look at the common variation. And maybe for this purpose, we could use a new hot topic in the arrhythmia research, polygenic risk scores, where the common genetic variation could be correlated with the phenotype of the patients. Maybe it's also interesting to look more into details in the epigenetics in Brugada syndrome, and this could give us some more information on the missing heritability of the disease. When it comes to iPSC cardiomyocytes, I believe that the focus should be paid to standardization of the differentiation protocols and improving the maturity of the cells. For this, I believe that because we are looking currently and focusing on the generation on a single cell type, maybe it would be interesting to study those models in a co-culture with another cell types, as a human heart is a composite of different cell types than only cardiomyocytes. And maybe with this, we are missing something to mature the cells. Moreover, as heart is a 3D organ, big potential lies in the search for 3D scaffolds and organoids to grow those cells in a 3D form. Whatever the course, I believe that we should focus on research of genetic background together with functional testing, as with this approach, we are looking at the full picture and not focusing only on a single piece of the puzzle. With this slide, I would like to thank everybody who was involved in this project, without whom I would not be standing here today. Thank you for all your support. Thank you to my promoters for reading the thesis and critical feedback on every chapter that I wrote. And thank you very much for listening, and I'm looking forward to answer your questions. Thank you very much.